You spoke at one time of the importance of the BLAST algorithm for your work and then further modifications of that. Could you speak a little bit about the analytical tools that you use and what we provide for that?
BLAST is a centerpiece of computational biology and genomics, a completely indispensable tool. I guess I would have to explain the acronym, which means Basic Local Alignment Search Tool, which may sound scary, but in reality it's an algorithm, and the program implementing it, which quickly compares sequences of nucleic acids and proteins. And there are, at the time of its development, which incidentally coincided with my arrival here,
Yes.
this was by far superior by any other available methods, and was actually the only real game in town, the only way to efficiently compare gene and genomic sequences. Very timely development. Since then, there have been lots and lots of improvement, both purely technical and conceptual, that make different flavors of BLAST suitable for particular purposes, as well as increase sensitivity considerably. Of course, I don't think that any other tool or method developed here really compares with BLAST in importance. Which, however, is not to say that no other useful things have been done. For instance, in my group, we developed so-called clusters of orthologous genes, COGS for short.
Could you repeat that phrase, class?
Clusters of orthologous genes. I'm
genes, yes.
using an obscure term here.
Yes.
But in reality, it means nothing mysterious.
Okay.
It simply means the same gene in different organisms.
Okay.
There is no really same gene in any pair of organisms. They change with time, but corresponding genes in different organisms. And then we speak about the same gene in, say, humans and mice. It's shorthand for ortholog, actually, for orthologous gene. So we developed methods. First, the idea itself of these cluster of orthologous genes, and then the algorithms and software to actually identify them. And this was important for, was and remains important for comparative genomics. And other developments here, such as, for instance, concern domain database developed by several people at the NCBI, which is also a very useful tool for understanding protein structure and function, and others. It should be noted, however, that there is substantial competition in the world now, much unlike 30 years ago,
Yes.
when very few people did high-level research in this area. Now there are many, and there is considerable competition that hopefully advances the field as a whole.
Well, that's also changing the way we develop and provide our services. So we used to be the only game in town where we had everything. We had the data, we had the analytics, we had the BLAST tools, and we developed the full stack. Now we're working more, especially under our comparative genomic initiative, the CGR initiative is designed to really make possible that connection between what's building outside of the NIH and the data that we have in the NIH, the annotation strategies people are using, the composition and comparison strategies they're using. So the library is changing as science is changing.
Yes, and I think it is very important. I don't think the challenge has been completely met, and indeed there are so many large databases around that the integration is a special task and not an easy one. by
Not
any
an
account.
easy one.